Host cell protein ELISA and HCP antibody coverage analysis. An ELISA, or enzyme-linked immunosorbent assay test kit, uses specifically designed and reactive antibodies to detect and quantify host cell protein, or HCP, impurities found in biopharmaceutical drug substances. The identification and monitoring of HCPs is critical to ensure purification process consistency and final drug substance purity. The capture and detection antibodies found in most ELISA kits usually only test for one or a few proteins at a time. But the antibodies found in the Cygnus HCP ELISA kit are different. They're broadly reactive and can measure hundreds of different HCP impurities in a single assay. But before we get too far ahead, let's walk through how this process actually works. The assay itself only consists of two main steps. First, add anti-HCP antibodies conjugated with HRP enzyme and a test sample containing HCPs or HCP standards to the well coated with anti-HCP antibodies. Cover and place the plate on an orbital shaker for 2 hours at room temperature. Then, wash the plate and do a colorimetric detection with a TMB substrate for 30 minutes. Afterwards, add a solution to stop the reaction between HRP and TMB. That's it. As you can see, the test does not require special expertise to run. It's also easily transferable, reliable, robust, and effective in high levels of protein. However, HCP ELISA assays do have some limitations. You'll need additional orthogonal methods to get a complete story, and the tests are only as good as the antibody used. Which brings us to the next part of the story, coverage analysis of anti-HCP antibodies. The wider the coverage, the more reactive the antibodies will be to HCPs in your cell culture process. The traditional coverage method is a 2D Western blot compared to a silver stain. This process shows coverage through counting the unique spots that appear in both the 2D Western blot and the silver stain. Unfortunately, this method lacks sensitivity and specificity. Cygnus developed a superior antibody coverage method called Antibody Affinity Extraction, or AAE. In this procedure, ELISA antibodies are covalently immobilized on a chromatography resin. Samples are then passed over the column packed with that resin to extract immunoreactive HCPs. The HCPs are eluted from the column, buffer exchanged, and concentrated to the original sample volume. The sample is then compared to HCP in the initial sample with a large format 2D gel detected by silver stain or 2D Dij, and an image analysis is performed to match spots. Comparing these results to the results found with a 2D Western blot coverage assessment shows both an increased sensitivity and specificity when using AAE. The column is also run under more natural conditions, so AAE is better predictive of how the antibodies will perform in the ELISA. The antibodies found in the Cygnus HCP ELISA kits have had their coverage assessment tested with both of these methods to ensure our kits detect protein impurities with a reproducibility that supports regulatory compliance. Have questions or want to learn more? Visit our services page and speak to an expert.